Welcome, everyone. Welcome to our MEPAN Meet the Researchers update. We're excited to have all of you here. As you all may know, it is World Mitochondrial Disease Week. So this is just another piece of the puzzle of awareness that we're able to share. So again, want to welcome you all to this interesting event that we have going on today, the UMDF and MEPAN Meet the Researchers. Now I'd like to introduce Sherry Breslow. Sherry is the mother to amazing Sydney Breslow, who's a MEPAN patient and the UMDF support ambassador. I'm going to let you know a little bit about that. For patient families that are on the call, we do offer support ambassadors for for many of the mito disease types, including MEPAN. And Sydney's willing and wonderful and able to help any family that needs help. So if you would like to be connected, please reach out to me you can find my information everywhere, but I will pop it into the chat. I'm Margaret Moore. I wanted to say a little bit more about the Breslow family. The Breslow family has been very involved with UMDF for a long time now, and they host a Cousins for the Cure event, raising money for MEPAN research. So now I'm going to turn the floor over to Sherry, and I'm going to go off camera. So take it away, Sherry.
Thank you, Margaret. Yes, my claim to fame and one of my greatest joys is being Sydney's mother. I want to thank everybody for being here for the first UMDF MEPAN webcast. A huge thank you to Kara Strittmatter and the entire staff and leadership at UMDF for their time, diligence, and persistence to make today possible. Also, thank you to all the researchers and clinicians for being here and for your work to try and help us find a treatment and/or cure. And lastly, thank you to Danny Miller for your help creating relevant questions for the panelists, for generating interest in MEPAN research, and for continuously reaching out to the MEPAN families and patients and their families. Dr. Douglas Wallace and Dr. Deborah Murdoch were the chairs of UMDF's Mitochondrial Medicine Symposium this past June, and they organized many scientific sessions that for the first time ever were focused on MECR and MEPAN. Dr. Murdoch presented what she and her team have learned from their MEPAN mouse model, and several other scientists presented the results of their latest research on how MECR functions in the mitochondria. Scientists, genetic counselors, and physicians gathered after the sessions for more extensive interactions about MEPAN, but unfortunately, due to geographical restraints, only a few MEPAN families could be there, and that is why we are here today. Dr. Wallace, Dr. Murdoch, and UMDF felt it was extremely important to have this webcast to share the exciting and groundbreaking MEPAN research that was presented in the UMDF scientific sessions, and to bring together patients, families, researchers, and clinicians in a virtual setting to connect with each other and learn about what is happening on the MEPAN research front. I want to personally thank Dr. Wallace and Dr. Murdoch for the MEPAN and other related research that they are doing in their lab, and for raising awareness in the mitochondrial community about MEPAN. And now please welcome our moderators for today, Dr. Douglas Wallace and Dr. Deborah Murdoch from the Center of Mitochondrial and Epigenetic Medicine at the Children's Hospital of Philadelphia.
Well, thank you, Sherry. That was a wonderful introduction. I really want to thank, again, the United Mitochondrial Disease Foundation for providing the resources, for allowing us to have this virtual meeting. As you heard, Kara Strittmatter has always been a great supporter and organizer for UMDF meetings, but Margaret Moore and Nicole Wilson are doing a great job joining us today on this Mitochondrial Awareness Week. The real organizers and leaders of this program are, of course, Sherry Allen and Sydney Breslow, Danny Miller and his two wonderful children and his wife. Again, the United Mitochondrial Disease Foundation that has provided all their resources to make this meeting happen. And of course, our Center and CHOP for support for these kinds of programs. As Sherry very nicely said, a number of the speakers that you're hearing today were ones that we had on June 29th and July 1 to try and raise awareness on MEPAN and to develop new research to find a cure. So we were really thrilled that UMDF would sponsor this virtual meeting so that many of you and the families could get to know the major leaders in this field and understand where we are and what we hope to accomplish in the coming years. So I would also like to mention that a little bit later, we will have Richard Kelly and his colleague coming on. They've been major innovators in the area of MEPAN and its analysis of patients with this disease. And I also would like to mention Yair Anexter from Israel, who was one of our speakers in June. Unfortunately won't be here, but he, together with Gali, who you're going to hear from, have been really leaders in the Middle East in this area. So what we really hope today is to have you, the families, become aware of what's happening, have you have an opportunity to ask any questions that you have, and allow the experts to give you the best insight we have on this field. So I'd like to then start with our first speaker, which is Gali Himmler, coming from Israel. And Gali is the senior pediatric neurologist and founder and director of the Sheba Multidisciplinary Center for Neurogenetics. And she really has been making a major effort in understanding MEPAN for a number of years. So Gali, are you on?
Hi, Doug. Yeah, thank you for this lovely introduction. And thank you, everyone, for inviting me today. It's a great opportunity to be able to talk to all the families since I wasn't able to attend the UMDF meeting. So thank you so much. Let me try to share my screen and let me know if... So do you see my screen?
Not yet,
I think the sharing was, yeah. Okay. Do you see anything now?
It's perfect.
Yes.
Okay. And do you see it in presentation mode, perhaps? If not, then never mind.
Perfect. It's perfect.
Okay. Okay. So I would actually, for those who are not familiar with the story of on how the MEPAN was discovered, I would briefly go over that first. So actually everything started with an 18 months old patient who presented to us during a prolonged illness with a dystonia chorea of the four limbs, along with a gait ataxia up to complete loss of ambulation. And now his history was unremarkable except for FTT and hypotonia with a little bit mild motor delay. But then when we started to examine his situation, the MRI showed bilateral T2 pallidal hyperintensities, which of course raised the suspicion of a mitochondrial disorder. Echo is normal as well as the eye exam and EEG.
Dr. Heimer, I think
Yeah.
I'm only seeing your first slide. Is that the same for everyone?
Oh,
Yes,
you're
thumbs up.
okay. So I will go out of presentation mode because do you see it now?
We're seeing your first slide.
No.
Oh, yes, now we see the patient. You just moved to
Okay.
the patient. Yes.
Yeah. So this was the patient. This was his MRI. So we'll just do it that way. Sorry. So the extensive metabolic workup was normal as well, except for a repeated increased excretion of 3-hydroxyisovularic acid in the urine. Okay, so I will just have to go down like that. Okay, so actually, exome sequencing and mitochondrial DNA analysis was negative. However, when we did a repeat analysis of his exome, we found 2 compound heterozygous variants in the MECR gene, which was at that time previously unrelated to any human disease. So the variants were predicted to be pathogenic, and the validation and familial segregation were supportive. So we followed our hunch, and he was put on mitochondrial cocktail along with Peptamine Jr. because of his FTT, and he showed a remarkable improvement of his symptoms. in parallel, we continued to explore the possible diagnosis of MECR as the causative gene, and then we used the gene matcher platform. This was supposed to appear at times, but never mind. So this is our patient, and then we posted these variants on the gene matcher, and sure enough, within a day, we got a response from Susan Hayflick. Do you see my cursor? So Susan's patient
Yes.
had a similar phenotype to our patient with a similar MRI picture. However, he was much older. He was in his 40s. So this reminded us of two other siblings that we've seen a few years ago who were also in their fourth and fifth decade with dystonia, optic atrophy, and a similar MRI. And we invited them in and we tested them for the three MECR variants that we've known on by that far. And sure enough, they had the same variants as the first patient. During the next few weeks, we were also contacted by the physicians of three other patients, one from Italy and two siblings from Australia. So at that time, there were 7 patients from 5 families over 4 continents, and we're actually pretty confident it's the same disorder. They all had a childhood onset dystonia and optic all but one, by the youngest, had optic atrophy, and they all had relatively preserved cognition, which is a little bit unusual for mitochondrial disorders. And of course, a similar picture on the MRI, as you can see the lesions in different parts of their basal ganglia. However, since, as I said, MECR was at that time considered to be unrelated to any human disorder, we had to go on and prove the pathogenicity of this gene. And then we teamed with Dr. Alex Castaniotis, who will be speaking later on. And he showed the functional pathology both in the patient's fibroblasts and in yeasts that carried the patient's mutations. So just a few words about the MECR. As you all know, it codes for the mitochondrial aryl-CoA reductase, which carries out the last step of the mitochondrial fatty acid synthesis, as you see here. Now, the empty fast, the mitochondrial fatty acid synthesis, is crucial for the mitochondrial function for several reasons. Its products affect the mitochondrial RNA synthesis and respiratory complex assembly. And the octanoic acid, the end product of the empty fast, is the substrate for lipoic acid synthesis, which is the cofactor of key enzymes in the mitochondrial respiratory chain. And if we go back to the empty fast pathway, then there are several disorders in humans regarding pathology in lipoic acid synthesis enzymes shown here, and usually the clinical phenotypes of these patients is much more severe than those of MEPAN patients. And one possible explanation for this could be that, as you see here, there is some endogenic source of octanoic acid as a byproduct of degradation of medium and long chain fatty acids, which is called beta oxidation. And this alternative endogenic source of octanoic acid might explain why the MEPAN patient have such a milder phenotype than patients with defects in LA synthesis enzymes. It also could explain why most of the MEPAN phenotype is related to the brain, which lacks fatty acid beta oxidation, in contrast to tissues like muscle and heart, which are commonly involved in other mitochondrial disorders. It could also explain the lack of classical mitochondrial biomarkers in most of the patients. And last, but most important, it could suggest a possible therapeutic approach with supplementation of lipoic acid or octanoic acid, which penetrates easily to the mitochondria. So since 2016, we've diagnosed and are currently following 9 patients within the Israeli National MEPAN Clinic in the Shiva Multidisciplinary Center for Neurogenetics. Now, we've established a MEPAN dedicated day. So during that day, the patients can perform all the following on the same day: the neuro exam, visual field and OCT test, neuro-ophthalmologic exam, orthopedic exam, and physiotherapy assessment. And in addition to the comfort of performing all of the assessments on the same day, this format also enables the patients to meet with each other during that day and to form connections, which we find very important. We know that repeat OCT tests show that there is a progressive substantial thinning of the ganglionic layer of the retina. We know that there is a considerable variability among patients, even within the same family, and we have a couple of siblings intact in most patients, so this is very optimistic. All of our 9 patients share the same 2 mutations. This has led us to study the prevalence of these mutations among Ashkenazi Jewish population, and as you see highlighted in red, there is an increased prevalence of these 2 mutations among this population. And lastly, which I'm sure Dr. Duta will speak about later on, is that we've shown a relatively low blood ferritin level in the 6 patients that we've tested so far that might be related to metabolism and toxicity. So we do also instruct our patients against using iron supplementation, even if their ferritin blood levels are low. So what are the symptomatic treatments for MEPAN patients? For dystonia, the main drugs that we use are clonazepam, baclofenazine, and trihexyphenidyl. Although Vivance, which is a prodrug of diamphetamine, showed anecdotal efficacy for dystonia in 3 patients who received it for their familial ADHD. Botulinum Botox injections can be useful for localized dystonia, and DBS is a possibility depending on the size of the basal ganglia lesion. However, I think that its outcome can be somewhat unpredictable, and you should be aware of that. For the patients whose phenotype is, of course, motor and visual aid should be adjusted for the patients, and augmentative communication devices are helpful for those with speech dystonia. For patients with all motor dystonia, they should go under evaluation and be consulted accordingly regarding their food consistencies. What about potential targeted therapies? So, of course, source of octanoic acid, vitamin E and C as antioxidants, and carnitine only for patients with low blood levels. And you see the doses here, either severe febrile illness, vomiting, or diarrhea cocktail, which can be given intravenously. However, alas, we do have patients on this cocktail that are still progressing. So we're considering perhaps additional vitamins, biotin, or like one patient who is receiving additional supplementation, and he's doing very good. What about idibenone, which is for LEM patients? And we have 1 patient years prior to coming to us, and he is doing motor or visual phenotype. So just going to my few last slides. So there's a lot of talk about AAV-mediated gene therapy, and I'm a great believer of this strategy for MEPAN. It's a promising therapeutic strategy in recessive disorders. It's a one-shot therapy, or good for at least 10 years, as we know now. We know that when it's injected to the brain, then there is a less risk of immune response. And due to its limited distribution, it's mainly effective for disorders that affect a relatively small area. And the 2 injection sites, which has the best results so far clinically, have been the bus, which we also have a patient who was injected with AAV, and various retinal disorders. And these are the 2 main affected sites in MEPAN. This is why I think it will be a good therapeutic strategy. However, there are the 2 main questions. First, will it improve current state or just prevent future insult? And if the second is correct, then how will we assess its efficacy? And natural history studies can be problematic due to the stochastic nature of the disorder. And this would highlight, again, the importance of the search for biomarkers for the disorder. So just before I finish, a few answers to questions that were sent to me from parents. First, regarding MEPAN and the immune system. So in answer to the question, we don't see increased tendency for infections in our patients. However, one must keep in mind that febrile illnesses increase the risk of metabolic crisis and additional neurological insult and should be avoided as much as possible. Regarding drugs that affect the immune dysfunction, like MS drugs, so I don't believe they would be beneficial for MEPAN, since this is not an immune disorder, it's in its core. About MEPAN and the mitochondria, so we've been asked about MAT, and which has been shown promising results in Pearson. However, since MEPAN, the MECL gene is a nuclear gene, and the proteins and enzymes that will be transferred with the mitochondria are predicted to be degraded rather quickly. So in the end, in the absence of intact gene to guide the manufacturing of additional intact proteins, I don't think this strategy will be beneficial for MEPAN. Lastly, they were asking about lipoic and octanoic acid supplementation, which I've already answered. And about the biomarkers, yes, we are pursuing the search for biomarkers. We've collected blood and urine from most of our patients, and also fibroblasts from some. We also have samples from patients with other mitochondrial disorders and non-metabolic disorders and as controls. We're still looking for funding to apply the metabolomics, but I hope that we will be able to ascertain some soon. And thank you again for inviting me and allowing me to speak with all the families today.
Thank you very much, Gali. This was a wonderful presentation. If it's okay, because of the technical issues, maybe we could move on and have questions at the end. Is that okay?
Yeah, sure. Yeah, and I apologize for the technical issues. Sorry.
No problem. So the next speaker is Deborah Murdoch, again, Associate Professor, Department of Pediatrics at Children's Hospital, Philadelphia.
Okay, can you see my presentation?
Yes, we do, but your pointer is faint. Could you go to the laser pointer option?
How's that?
Much better.
Okay, great. So my name is Debbie Murdoch. I am a scientist at the Children's Hospital of Philadelphia in the Center for Mitochondrial and Epigenomic Medicine. I'm really a basic researcher, and so I come at this all from a very different point of view. And so I thought I would take some time to just explain how it's useful to study MECAR and MEPAN in other model systems. There we go. So at the center, at the CMEM, we really work on all things having to do with mitochondrial energy production, and that is primary mitochondrial diseases all the way up to how the mitochondria play a role in COVID infection and that kind of thing, and the immune system. And so really the big picture here is that mitochondrial disease is energy deficiency. And most of those original mitochondrial diseases that were found were in this machinery down here that are used to take the food that you eat, and then molecules go through this machinery and you end up with ATP, which is energy. So if anything's going wrong here, you're going to have a problem with energy in your tissues. So a lot of those original mitochondrial diseases were defects in complex 1 or complex 3, and we could sort that out. And like I said, these are all proteins that have been mutated in the past. And what we're talking about here is something a little different, because there's this other pathway going on, we know now, and that's mitochondrial fatty acid synthesis. And mitochondrial fatty acid synthesis doesn't make proteins like this, it makes fats. And, you know, the bigger group of fats is like avocado oil, butter, all these things. The oleic acid that you just heard Dr. Heimer talking about is a fat. And so that's the question, is how these fats play a role on energy dysfunction in the mitochondria. So I think there's a lot of people working on that in a lot of different ways. So all of these model organisms have been used to study mitochondrial fatty acid synthesis. So all the way to yeast, which are single cell organisms. And of course, we use these for making beer and making bread, but they have mitochondria and they have mitochondrial fatty acid synthesis, and they have their own version of MECAR. All the way up to the mouse. I don't know if this is up or down, but the mouse also has mitochondrial fatty acid synthesis, and you know, the defects in the mouse are tissue specific, just like they are in the humans. So we've learned a lot from all these organisms, and I'm going to tell you why we are using mice for what we're doing now. I should also say that most of what we know about mitochondrial fatty acid synthesis comes from studies in yeast and fungi. So those have been really important studies, or when we got the first mutations in patients, we wouldn't have known what we were looking at all. So this has been very helpful, this historic work. So we've decided to use the mouse for our systems. There are benefits to it. Of course, mice have all the same organs pretty much that humans have. They have the brain and the eye, which we know are absolutely affected in MEPAN. They also have muscle, comparative muscle, and we can also look at the muscle and see if it's affected. And I think everybody would be interested, patient families would be interested to know if muscle is really directly affected or if it's all downstream from the brain. And they also have an immune system that's very similar to the human immune system. So there's the basic immune system that all organisms have, Drosophila have it, but then there's T-cell based immune system. And we know that mitochondria affect the immune system too. And so we think all of these things make the mouse a really good model for study. The other benefit about the mouse is that if we want to test therapeutics, we don't have to go directly to the humans. We can take a mouse model of MEPAN, test any kind of therapeutic, a drug, a gene therapy, and see how it works first in the mice. And that's a lot safer way to do things. And it's a lot, you know, we'll be much better at clearing things through the FDA if we have mouse studies. So we set out to make a mouse of MEPAN that has the same kind of mutation that the patients have. And you might all remember that most of the time in MEPAN, as Gali was saying, there's a mutation on one chromosome, maybe from the mom, that's just one point mutation, and it changes the protein a little. And then usually there's a stop codon on the other chromosome, maybe that came from dad. So it's this mix of things that seem to be happening in most patients. And what we've ended up with, we tried making all three of these specific point mutations. This is the only one we were able to make in the mouse. in our lab. And so what we have are 3 different mouse models that all have this mutation on one chromosome and a stop on the other, but they're all different. So this is just a representation of those 3 original mice. They all are a little bit different. There's one of them that is a little smaller than the others, and it doesn't live as long, but the other 2 have absolutely normal lifespan. And in the cage, they're fairly hard to tell them apart unless you challenge them to do something. So if we want to use these mice for therapeutics, first we need to show that they really are what we think they are. They have mutations in MECR, and then we want to show that they actually have defects that look like MEPAN patients. So here you can see this is the brain of a mouse. The NIH has put a lot of money into mapping the brain of the mouse, and so we can kind of tell where things are, different parts of the brain. And you can see for orientation here, you know, this is the mouse. Ours look more like this. Mice have this huge olfactory bulb that let them smell right here, and that's what this is. And then here's the cerebellum that does a lot to control your balance and your movement. And in this picture, this is just a regular mouse that has been stained for MECR protein. So anything brown is MECR. And so, you know, you would ask, does that help us learn anything about MEPAN? Well, the idea is that anywhere that MECR is expressed should be important in its function. And that has borne true because when we saw the olfactory bulb was so strongly stained, we tested smell in the mice. And we can tell that they are not able, almost unable to smell anything. So this is only in the worst model. The other 2 models have a little bit of a deficit in smell, but that's just our indication that where MECR is going to be an important place in the brain for the disease. And then this is a wild type brain, a normal brain. And then this is our mouse mutant model. So MECR or MEPAN is, or MECR is almost completely gone in the mouse brain here. There is some left. You can see some residual brown staining, but you can tell we pretty much eliminated MECR from the brain. And as I said, in this model, we can also look at the eye. And this is the retina of the eye, which is, if you're looking at your eye, there's a back part of the eye that your ophthalmologist is always looking at. It's the back part of the eye with all the neurons back there that detect the light. And in these pictures, this side is a wild type retina, a normal retina stained with MECR, and it's in the mitochondria. These are all the places that you would see mitochondria. They're also stained brown with MECR. And in this case, this is the knockout, or the point mutation. And we really lost a lot of MEPAN or MECR in the retina. But does that cause a phenotype? Oops, I lost that. Okay, so here's the retina stained with a protein that comes on when you have stress in the retina. And the wild type mouse doesn't have this kind of staining, or sorry, this staining, it only has this. This means that the retinas are stressed, and that's our first indication that the retinas are dysfunctional in these mice. We're also doing OCT, as you heard they're doing in the patients, and we'll be able to see if the retinas are thinned in these animals. So therapeutically, we have something that we can look at here, and if you give a drug, can you make the staining go away? This is just an example of how gene therapy works in the eye. We've been working on several mitochondrial diseases that cause blindness, and we've been working on gene therapy in those systems. And this is a normal eye. It's a mouse eye that's uninjected. In this one, we've injected a gene therapy vector into the vitreous of the eye. And you can see again that this protein, it's not MECR in this case, it's something else, it's catalase, is targeted really darkly into the mitochondria. So we know we can get a gene in there. Is that something we should be trying in the MECR mouse? Okay, now I gotta go back to what I skipped. So then, do these mice have a movement disorder? So we spent a lot of time characterizing their movement, and here's one of the tests that we do, and probably the one that's easiest to measure and see if therapeutics are working. So this is an example of this guy has to have pretty good balance to be able to stay up on this log in the water, especially if this guy's spinning it. And so we do something similar in the mice world. And you can see over here, they have a rod that's like that log in the water. We put the mice here, and then this thing starts spinning and going faster over time. And you can see that they'll drop off the log when they can no longer balance up here. So this is the performance where we get numbers. So this is how long it took them to fall. This is how an unmutated mouse behaves on that over several trials. They get a little better over time. The worst of our mutants falls off of the log pretty quickly. And so you can imagine this is a before and after treatment test that we can do. So you can treat a mouse for a certain amount of time with whatever you want to try, lipoic acid, that kind of thing, and see if it improves the score. And so between that and the eye, we have two of the main functions of MECR that we can test. And so this is just kind of an overview of the people that are in the center. Of course, Doug Wallace makes everything there happen in environment and cash and everything else. These are a bunch of different people that work in the lab, including these two who are really the mouse whisperers. They take care of the MEPAN mouse. And they're also terrific neurologists, so that's really great. Transgenic core helped us make the mice, and the neurobehavioral core at Penn helped us test all of these things. So I'd like to thank the patient families, because I think we have learned so much from knowing the people who are dealing with this dysfunction, and we know what to look for in the mouse, and hopefully we can get to a place where we can do something that's really improving quality of life for people that have MEPAN. We'd also like to thank the Breslow family, the lovely Breslow family, who we really enjoy working with. And we'd like to thank UMDF and Cousins for a Cure for funding some of this work, and our NIH TR01 that funded some of the early work. So, and thank you all for listening.
Thank you very much, Debbie. If it's all right, again, since we're falling a little bit behind, we'll maybe save all the questions for when all the speakers are done, if that's okay. We'll move on to Alexander Castantios, and he is on the faculty of biochemistry and molecular medicine at the University of Oulu in Finland, and has been interested in MECR for some years. So, Alex?
Yes, okay. I've started to share. Now I'm going to put my presentation to presentation mode. Can you see that?
We see your slides very nice.
Okay. All right. So this presentation was kind of initially targeted to a different audience. I apologize. I had to throw this together in half an hour now because I wasn't aware I had an opportunity to talk, but I think this would be okay. So maybe to recap, as Debbie said, so mitochondria have very important roles in eukaryotic cells, and the most prominent one that we know about is providing the energy that we need to maintain the cell. But there's other roles actually also. And let me get a better laser pointer. Mitochondrial fatty acid synthesis is a process in mitochondria that hasn't been sufficiently well characterized, but what we know is very important for mitochondrial function. And mitochondrial fast didn't pop onto this stage just a couple of years ago, but actually it's there's over 30 years of history on the research of these components, and we've been involved in this over the past 23 years. Actually, we haven't had a presence in the US for the last 10 years, first because of funding reasons and because of COVID. So it's actually great to have this forum to talk about our work. The pathway is highly conserved from humans to Baker's yeast. Debbie mentioned that also. So it's virtually identical from these simple yeast, like one cellular, single cellular organism, to humans, there's very small differences only, and the human protein basically can fix mutations in the yeast if we introduce it into yeast. And this makes a fine model. As was mentioned before, the pathway produces octanoic acid to generate lipoic acid. That's by now very well established. But there's evidence that there's another fatty, actually, I don't know if it's just one, it might be several different longer fatty acids that have an important function in controlling mitochondrial protein complexes. And we've shown just recently that, and this is in the yeast model, that just to be able to make lipoic acid in mitochondria is not sufficient to support mitochondrial function. So this is a paper from the beginning of the year. You see, we have a mutant that makes near wild type levels of lipoic acid, but it cannot respire this yeast mutant. It actually does worse than a mutant that has lost one of the mitochondrial fast enzymes. This is a graphic depiction of the pathway. Again, this is more now the mammalian one. There are several different steps. Each of these steps in the synthesis pathway is catalyzed by specific enzymes, and each one of these enzymes, if they're not present, it would be predicted that they would cause a deficiency that's very similar to the case when we have a MECAR deficiency. So there would be only small difference in terms of what molecule is attached to the acyl carrier protein when actually the whole synthesis happens. Now, if there is a defect in mitochondrial fast, and so you don't get this acyl carrier protein with the appropriate fatty acid chain attached, then you have problems with getting sufficient amounts of internally synthesized lipoic acid. But I think what is a very important feature is, and that wasn't predicted initially, is that you have problems with assembling complex 2, complex 3, complex 1 in mammals. In yeast, we know actually also complex 4, and the respiratory chain is not properly assembled. And it should really be mentioned that this acyl carrier protein with a fatty acid attached that was synthesized by the pathway. This is contributing to the function of the iron sulfur cluster synthesis machinery, which makes a very important cofactor that affects not only mitochondrial function but general cellular function, and that should not be neglected. So we've been studying mitochondrial fast for a long time, initially from a purely academic interest. There was this pathway that was poorly characterized, but clearly had an important function. So we just started to look at this initially in yeast, and we characterized several of the enzyme components, and as well as components of the lipolation process. As I said, there's a longer fatty acid that is required to actually, that is made by the pathway that's required for proper mitochondrial function. What we've been able to show, and so this was, we started this project before the patients were known. We had a mouse model where we could knock out MECAR completely, and if you have a complete knockout, the mice die very early in development. But if you do this specifically in Purkinje cells, you see that you get neurodegeneration, and this the effect that you see from this neurodegeneration is that the cerebellum is smaller, and after 10 months after the knockout, so we can do this at a specific point. This happens at a specific point in the mouse that is already born. So it's not when the mouse is born, the knockout is not yet happening. It happens 3 weeks later. If you get this knockout, you completely lose the Purkinje cell. So this is wild type. Oh, and this is 9 months. This is the mutant that doesn't have MECAR in the Purkinje cells and completely loses it. So this is a formal demonstration that you need mitochondrial fatty acid synthesis in order to maintain neurons. Now, so maybe I can go quickly through this because we've heard this. MEPAN is a hereditary disorder, early onset. there's specific changes in brain structure that Gali talked about, and you see the progressive dystonia and optical atrophy. And there's other mutations in the FAS, mitochondrial FAS pathway that have been either reported already. We know about a mutation in the ketoase reductase that has not been published, but these have been identified, and then they do look similar to MEPAN patients. Now, we also set out to make a mouse model, and so this is from Gali's publication from 2016, where the first patients were described and the different mutations. So this is a depiction, like a graphic depiction of the protein in a linear way, and you see where the mutations are. Now, Debbie talked about the mutation here. So this is number 5. What we were targeting was this mutation, so the glycine 232 glutamic acid, which is the one that was found in the Israeli patients. And so here you can see where this is located in the protein. So this is a dimer. This is 2 times the same protein, like our protein that makes a functional enzyme. You see it's here actually on the outside of the protein. The mutation causes a steric clash. It probably destabilizes the protein. We haven't formally shown this, but all the evidence that we see points to that. So, whoops. Yeah, so this is from the original publication where these mutations were found. And typically, and this was also mentioned, they're always, you have the point mutation, which gives a partial function, so it's not a complete loss. And then you have another mutation that either gives a stop codon, or there's a problem, like this is an insertion. So it basically makes a completely non-functional protein. So always this combination of partial function and a complete null mutation. And we showed this actually, and this is also where yeast again came in really nicely to show this. So you see the glycine 232 glutamic acid mutation. If you test this in yeast, you see it has a little bit of a function. So this is growth on a non-fermentable carbon source where the yeasts have to respire. So they need functional mitochondria, and it does a little bit, while this C855 T2G mutation is a complete null. So you can see there is some function left in this glycine 232 glutamic acid mutation. And this was shown by Gali already. So we were able to show in this publication that microprotein fails to accumulate in the patients. And therefore we thought this was maybe a good mutation to model, because it is very, very highly conserved. You can see this from humans, and so those are all mammals. This is C. elegans, but also yeast. So this is a place in the protein where this glycine apparently needs to be there to have a properly functioning protein. So this seemed like a good target to go for. Also, I think maybe it was because this was one of the first patient that was characterized. We kind of focused on that. And we got this mutation by doing CRISPR-Cas9 genome editing. We were apparently very lucky that we did get the mutation. Now, from the beginning, we set out to only model the point mutation. And because we had already the knockout mouse, and so in the knockout mouse, you basically have a MECR variant where one of the exons, so one of the parts of the DNA that encodes the protein, was actually removed. So that already made a null. And our MEPAN model mouse so has the glycine 232 glutamic acid mutation allele on one chromosome, and it has this knockout allele, so missing the entire exon 2 on the other chromosome. And so did it work? So unfortunately, I don't have as much data as Debbie. I haven't been able to. We have some his histology, actually quite a bit of histology data, but I just couldn't get to it right now. It's evening and nobody's here anymore. They couldn't get the data, but we were basically also able to show that in these mice, this is liver MECR accumulation is strongly reduced. We actually also looked at brain and we had the same result. And the Rotarod test was introduced by Debbie. And also here in our mouse, we can show that there's a clear deficiency in how much time they can spend on the Rotarod, as well as the grid hanging test. So this is really literally a metal grid where the mice are holding on to, and they fall off much earlier in this case. So I wish I could show you more data, but we do have some. I might be able to talk about some of it at least. But as my kind of final slide, mitochondrial Fas is a highly conserved mitochondrial pathway that coordinates mitochondrial function with cellular metabolic status. We've suggested that, and then Sarah in Rutter's lab actually showed really nicely that the compound or the component that does this is acyl carrier protein, is an acylated form of acyl carrier protein, so ACP that carries a fatty acid. This function leads to neurodegenerative disorders, and we've modeled this mutation. And we think this mouse is a suitable model and might be a nice, I think it's a nice additional model of to what Debbie has already shown. Yes, and this is just the people who've done the work and the different organizations that have supported us in the work. Yeah, thanks for giving this opportunity, and apologies for this. Could have been better if I would have had more time to prepare, but I hope this will help a little bit in understanding.
Thanks, Alex. That was a wonderful job, and thank you for coming up with your presentation at such short notice. If it's okay, because time is moving on, we could hear from Sarah Nowinski, who is in the Van Adel Institute. She's a faculty member there and did some really seminal work with Jared Rutter when she was a postdoc, and is now continuing this work as a faculty member.
Okay, everything looks okay with the slides?
Perfect.
Great. So thank you for that, and thank you for the invitation to be here and talk to you all today. Like Debbie and Alex, I'm a biochemist, a basic scientist, not a clinician. But what I'm going to tell you about today is actually another patient with a mutation in the MitoFAS pathway, not in MECR, but in the first enzyme in the pathway that attaches a new malonyl-CoA to that acyl carrier protein, and that enzyme is called MCAT. So like Alex, I was working on studying the biochemistry of the pathway, how the pathway is functioning in cells in the lab. But we published a paper, and I was approached by a geneticist named Dr. Bryn Webb, who at the time was at Mount Sinai. She's now at University of Wisconsin-Madison. And so she had a patient who was a 2-year-old male at the time. And the patient had been born prematurely at 30 weeks because the mom had preeclampsia and was induced because of the preeclampsia. So the child had been diagnosed previously with failure to thrive and retinopathy of prematurity, and was being followed up by a social work team, also had some nystagmus, which is eye jerking. But the patient came in, as many do, following infection with a metabolic crisis. And at that time, so this was when the patient presented to Dr. Webb, he had notable developmental delay, only spoke 2 to 3 words as a 2-year-old with no 2-word phrases, could not walk independently, as you can see, was still very small in size. And so Dr. Webb's team started following up on this patient and found some abnormal MRI findings, plagiocephaly, hypotonia, and decreased muscle mass. And so this led them to a differential diagnosis of mitochondrial disease. because they thought it was unlikely at this point that all of these symptoms were simply following on from the premature birth. And so whole exome sequencing revealed a variant under suspicion in the mitofast gene MCAT, and this is a threonine 271 to isoleucine substitution in MCAT. Like other mutations, this was thought to be homozygous recessive, but the patient is homozygous for this point mutation. It's not a compound heterozygote like the other mutants that we've been talking about so far today. So Bryn got in touch with me and we started looking at mitochondrial function in these cells from the patient. And what we found is that in both lymphoblasts here on the left, as well as fibroblasts here on the right, the patient had reduced levels of ETC complexes 1 and 4 here on the blot, strongly decreased levels of the MCAT protein itself, but no reduction in lipoylation. And you can see that here in this P lane. And this is the first, to our knowledge, description of a patient with a mutation in the mitofast pathway that doesn't have a protein lipoylation deficit. So how could this be? What is going on here? Well, Alex just introduced that mitofast makes more than just octanoate, which then is further converted to lipoic acid. But there's also good data in yeast and in test tubes that it can make these longer fatty acids, so longer lipid chains out to 14 or 16 carbons. And Alex kind of alluded to this, but the identity of these longer fats and what they're doing in cells is still not really well understood. But what we know from our work is that those longer fats are really important for some physical interactions between proteins in the mitochondria. And so we found that this acyl carrier protein has to be able to touch and bind and physically interact with a family of proteins that are called the LYRM proteins. Oh, this slide didn't work correctly, so I'll just jump ahead to this one. So what we found is that physical interaction, the being able to grab and hold onto the LYRM proteins, it actually requires a fat, one of those longer fats, to be built on the acyl carrier protein. And when mitofast is mutated, it can't build those longer fats on the acyl carrier protein. And so it can't grab and hang on to these LYRM proteins, and they are degraded. Now, the reason that's important for mitochondrial function is that these LYRM proteins are electron transport chain assembly factors. So the electron transport chain, right, it's all these big complexes. They have a lot of subunits. I think there are 93 different genes that encode different pieces and different components of this electron transport chain. And so they have what we call molecular chaperones. And it's kind of like a chaperone in real life, right? They're gonna make sure that this assembly process proceeds as it's supposed to. And when you don't have the chaperone, things don't turn out right. And so what we were able to find from all of this work is that there's really these two functions of mitochondrial fatty acid synthesis in cells. One is lipoic acid, which we've spent a lot of time talking about today. But the other is that these longer fatty acids are required for building a fully functional electron transport chain. And they're actually also important for the synthesis of some cofactors that are really important for electron transport chain function as well. So I didn't go into a lot of the biochemistry supporting these cartoon models, but what my research has really shown is that you need both of these functions of mitochondrial fatty acid synthesis to have a healthy mitochondria. And this MCAT patient is the first demonstration in a human patient that some mitochondrial fatty acid synthesis mutations can drive pathology in the absence of protein lipoylation defects. So if we're only looking for lipoylation as confirmation of variants of suspicion, we may be missing patients. And then also, Alex kind of alluded to this as well, that these are two separate functions of mitofast in humans. We know that if lipoylation is intact, or if you only rescue lipoylation in a setting where both products are missing, it's not enough to have a healthy mitochondria. And so I think that it's important as we think about treatment strategies for patients to take this into account and not just be solely focused on rescuing lipoylation. So like everyone else, I have my thank you page. I just started my lab at the Van Andel Institute 2 years ago, and this is the group of people that are doing the work here now. I also want to thank everyone in my lab in Utah, which was where some of the foundational work was done. My collaborators, I mentioned Dr. Webb is the geneticist who's now at UW-Madison, as well as the patient and their family. The interesting cases are really sometimes what helps us learn a lot about how pathways are working and what's going on. And then I would also like to thank all of my funding sources. So thank you.
Thank you, Sarah, for that really exciting presentation. And again, sorry, but we probably should move on. Our next speaker is Jay Lee Steiner from Vanderbilt University, and she works with Jeff Mathmel, who is a professor at Vanderbilt. And now we're going to hear from her.
Great, thank you. I will share my slides. All right. So hi, my name is Kaylee, and I'm a graduate student in Jeff Rathmel's lab at Vanderbilt. And so like Sarah, I'm also on the basic science side, but I really hope to convey of how MECR is important in T cell function. So what our lab studies is studying the metabolism of T cells. So we come from a very basic science approach. And what our lab has found is that different types of T cells, such as cytotoxic T cells, which directly kill cells, or helper T cells, which can help activate other cells, they rely on different metabolic programs, and this can influence their function. So once T cells can get activated, they utilize different processes, such as they can increase glycolysis, increase the TCA cycle, and they can also increase such as lipid synthesis for a dividing cell. Now, what I talked about, different subsets of T cells require these different programs, such as T regulatory cells, which are cells that can directly suppress your own immune cells, rely on fatty acid oxidation, compared to effector and pro-inflammatory T cells can rely on glycolysis and utilizing glutamine. So our lab tries to utilize these functions to change the function of immune cells. And so we want to identify molecular targets and metabolic mechanisms to modify these immune cell fates. And this can really be important in such as cancer, to change and increase the function of T cells. You don't want as much T cell function. So in our lab, we have utilized CRISPR screening approaches, where my project started is taking genes involved in lipid metabolism, such as lipid synthesis and actually mitochondrial fatty acid synthesis, And we can individually in a cell knock out this gene in our primary T cells. And we can put this in an in vivo model to see which genes will be selected as important for T cell function. And when we do this, we can then, after disease development, isolate back out organs of interest and really compare which genes are enriched or depleted, meaning which ones have a fitness advantage or disadvantage. And so briefly, what my project has looked at is, one, we've done these CRISPR screens in many different models, such as a lung inflammation model, an inflammatory bowel disease model, and a cancer immunotherapy model. We consistently see MECR coming up as a significant hit of significantly depleted, meaning that the normal function of this protein is important for cells, and when they lose this, they don't function as well. Interestingly enough, OXS-7 and MCAT, which are also part of this pathway, also came up in these screens. So we know that on the clinical side that mutations of MECR can cause MEPAN, and this is important in changes in inflammatory diseases and can also cause inborn errors of metabolism. And our lab has also focused on studying its role in T cell function. However, with my project, we're looking at the basic science side of the questions of my project is, how does MECR impact T cell function in these different inflammatory disease settings, and how does MECR change T cell metabolism? And so for just a graphical abstract of what I have found with my project is that I have looked at using CRISPR-Cas9 to knock out or have MECR non-functional and see how it impacts T cell function. What I've been able to see so far in vivo models is that it can decrease the production of interferon gamma, which is a pro-inflammatory cytokine, as well as decreasing TBET, which is a transcription factor for a subset of T cells. I've also found that by knocking out MECR, it can increase apoptosis, which is a form of cell death. Also, as people have talked about, they have seen that the loss of MECR causes some complexes to be non-functional. And I would also hypothesize that this would be the case. And possibly if all these different processes can explain why we're seeing a difference in T cell function. That possibly if the electron transport chains aren't intact and not functioning, or lipoic acid synthesis is not being done properly, that can influence T cell function. And so just for some of the questions that I have gotten before, to just end quickly, is how is MECR involved in regulating certain cells of the immune system? That's a very good question, one I'm trying to answer with my project, that we think that the lipoic acid and electron transport chain complexes are really important in the natural function of T cells. For the second, I'm not a doctor, so I really don't know about the disease pathology if it includes immune dysfunction. What we would like to do is collect blood, possibly from patients, and analyze the immune cells. And even if there's no difference in the total or percentage of T cells, there could be a difference in their T cell function or metabolism, which would still be interesting and can really tell us some things of what's happening in the immune system of these patients. The third was already kind of answered by previous talk, but I really don't think that other autoimmune disease treatments would help MECR or MEPAN patients, and that are patients with MEPAN more susceptible to viral infections and COVID. I'm not a doctor, so I also don't know, but I do hope in my studies to test this. We do have currently going a mouse model, which will have the loss of MECR, specifically in T cells. And what we can do is challenge these mice or infect these mice to see how well they clear these infections. And so that's the end of my talk, and I can take any questions too once we're all finished and everything. So thank you.
Thank you very much, Kaylee. That was excellent. I wondered if Dr. Deb D. Duta with Hugo Bellin wanted to comment something as a part of our question and answer period.
You're muted, Deb.
Oh, sorry. Yeah, I'm open for question answer, as well as I can take you through my slides briefly if needed.
Why don't you do that quickly then?
Yeah, sure. Can you see my presentation?
Says it's starting, but we don't. Oh,
Yeah,
okay.
so I'll not go with this detail of the patients, but I'd like to start with these two cases. Like, these are from Danny's kid, and they were enrolled in the UDN program, and we work in the UDN program. And therefore, we started to model this in flies, and our study is mainly focused on flies with some sort of evidence from human fibroblasts as well. So as we know all, that MECAR is the last step. MECAR is the enzyme that is required for the last step of mitochondrial fatty acid synthesis, and that produces acylated ACP. So these are the fatty acyl chains that is attached to ACP. And you can imagine if MECAR is not there, those acyl chains are not produced. But how does it cause neurodegeneration? For that, we used fruit flies, and you can see we used fly mutants using different strategies, and I'm not going into the detail of all these, but I'll briefly show you what we did. So we made a CRISPR knockout allele of this MECAR in flies and found this is lethal at developmental stage. In that background, we used a humanization strategy. By expressing human MECAR, it fully rescued the lethality. But when we modeled the point mutation variant, it either failed to rescue or only partially rescued the lethality, indicating that these patient variants are loss of function in nature. But we were more interested to understand how is it causing neurodegeneration, how MECAR affects neuronal functions. And therefore, we performed electrophysiology. And this is a typical ERG trace that gives you an idea about the synaptic transmission and phototransduction process of the neurons. And when we did this experiment in the mutant clones, in the young flies, we did not see any kind of differences. But when we did it at old flies, 15-year-old flies, you can clearly see that the synaptic transmission and phototransduction process is impaired. Also, when we perform transmission electron microscopy on these neurons, we found that the mitochondrial structure morphology is well affected. And consistent with that, the mitochondrial function, as indicated here, like with the ATP levels or the TMR staining that indicates mitochondrial membrane potential, another important functional marker, they were all down. And at this stage, we took patient fibroblast and measured different respiratory parameters like the ATP and different respiration. And they are all down, indicative of mitochondrial function is impaired. So it led us to the question that what caused this kind of defect in mitochondria? Since MECAR is involved in fatty acid synthesis, we thought that decreased lipid synthesis is the cause for this. And lipids are very important for mitochondrial structure and function. So to test that, we performed lipidomic analysis. And contrary to our expectation, we found a special class of lipids, sphingolipids called ceramides, which are very important for the membrane function and often implicated in neurodegenerative disorders, are increased in the patient fibroblast as well as in our mutants. And therefore, we were interested to ask the next question, that how does loss of MECAR cause this ceramide accumulation? So there can be multiple reasons for ceramide accumulation in the cell, but we focused on iron sulfate cluster synthesis. Why? Because there is another pediatric neurodegeneration called Friedreich ataxia. And in that disease, iron sulfate cluster biosynthesis defect is known to cause iron toxicity. And we and others have also found that in these patients, there is increased ceramide levels. That makes a connection that maybe iron toxicity and ceramides are connected. On top of that, if you look at the complex that produces these iron sulfate clusters in the mitochondria, there are 5 proteins. One of them is frataxin that causes Friedreich ataxia. Another one is the ACP, and this is the ACP which is acylated with longer fatty acid chains. And therefore, we thought that if you lose this acyl chain from 16 carbon to here in the MEPAN mutant, it can be maximum 4 carbon. So it may affect the assembly of this complex, and that can hinder iron sulfate cluster synthesis, leading to iron deposition. So we test first the iron levels in the fly mutants, and this is the mutant brain. You can see clearly that there is iron deposition in these mutant brains. Also, we validated it with another independent method, and we found that indeed the iron levels are increased. And when we shared this with the GALI, we came to know that the patients who are having iron ferritin levels, they are at the lower end. And the 2 kids in the UDN, they were having the lower ferritin throughout. So it is possible only if there is iron sulfate cluster synthesis defect. And therefore, we tested that. So we basically looked at an enzyme called aconitase. This is in the mitochondria, and it requires iron sulfate clusters. If you lose iron sulfate clusters, it affects its activity. And this is a marker, and therefore we looked at the aconitase activity in both patient fibroblast and the fly mutants. And it was indeed reduced. So at this time, it was interesting for us that the iron was increased, the ceramides were increased. So are these 2 things related? Or if we reduce these levels, can it help in the phenotypic rescue? So for that, we went for another strategy. And here we actually performed neuronal knockdown of MECAR in the adult flies. And as expected, young flies were not that affected, but 25-day-old flies, they were having locomotor defect. And we tested different genetic as well as different drugs to feed the flies, and we saw rescue. But here I'm going to show you 2 key data. And in that, we fed these neuronal knockdown, MECAR neuronal knockdown flies with deferriprone that reduces iron, and desipramine that reduces ceramide levels. And in both cases, we saw that there is a kind of nice rescue in the phenotypes in locomotor activities. And also we worked on these. We looked at the ceramide and iron level, and ultimately it seems that the desipramine cannot reduce the iron level, but deferriprone reduces the ceramide levels. So overall, I think that in the MEPAN patient, also in the MECAR mutants, there is iron toxicity due to iron sulfate cluster biosynthesis defect. And this iron metabolism defect triggers the ceramide levels, and that leads to mitochondrial abnormality and neuronal demise. And this is the summary of our work. And with that, I would like to thank Hugo and all my collaborators and funding agencies and the patients and families for their cooperation. Thank you. I'm open for any question.
Yeah, thank you very much. Is Dr. Richard Kelly on now? Did he want to make a comment? Okay.
I just moved him to a panelist, so he'll be able to respond now.
Okay, Richard.
Can you hear me?
Yes, very well.
But you can't see me. Oh, okay. Do I have any comments? I think one thing that for those who are not living in the MECAR world, one of the strongest arguments I thought for the importance of the long chain fatty acids is that the KMs for MECAR decrease with chain length. So going from C8 down to C16, it's a substantial drop, which would mean that there's increasing affinity, and the Vmaxes, as I recall, are not that much different, but it's just circumstantial evidence that long chain, that a goal of MECAR, or a goal of the mitochondrial fatty acid pathway, is to produce long chain fatty acids, and that the lipoic acid is just a sort of a side project of the enzyme. So yes, I think that there's a lot to be learned about the effect or the consequence of the deficiency of long-chain fatty acids, and looking for ways of ameliorating the problems secondary to that. So all these different, it's a very complex, phospholipids in particular, a very complex system. And I've invited Miriam Greenberg to join because she's an expert on phospholipids in mitochondrial function and has done a lot of work on Barth syndrome. So I personally think this is a very fruitful area of investigation for MEPAN.
Thank you, Richard. Richard has really been the pioneer in the clinical workup of these patients, and we're really appreciative of all his important contributions. Dr. Greenberg, did you want to mention something?
I'm not sure she's on.
Oh,
She had a conference that might run late, so
okay.
she
Well,
may
that's
not
fine.
joined.
So we have two questions that are on the question and answer from Mr. Mike Cohen. He would like to, Mike Cohen has a cousin in Philly, and he's I think age 55, wants to be seen. The mitochondrial medicine clinic, Matt Duran, can see MEPAN patients. He's seeing some. So if you'd like to email us, we can put you in contact with him to be seen. There were several questions that were raised, but nobody seems to have raised their hand. So if people could raise their hand, then we can call on you to get your question, please. Okay. Lisa Zent, do you want to ask a question, please? Can you? Nicole, can the individuals have access to the?
They can. Leslie just needed to unmute. I believe she's
okay.
ready now.
Great, great.
Sorry about that. I have several different diagnoses that I'm realizing could be all just MEPAN, but would the treatments be different?
So, good. Would Dr. Steiner or Dr. Kelly wish to address that question?
Can you ask a more detailed question? I wasn't sure if I understood.
I have asthma and Crohn's, fibromyalgia, and a whole host of different things, Meniere's. And I'm wondering, instead of treating those as individual illnesses, can I look at it and treat it as a MEPAN diagnosis? And if so, would that treatment be different?
So are you saying that you have MEPAN, or you think that because—
No, I do have MEPAN. I definitely have MEPAN.
Yeah, you do.
Mm-hmm. Yes.
Well, yeah, this has to be looked at in terms of the individual things, but suffice to say that as described, mitochondrial function is very important in the immune system, and the largely, and it's not unique to MEPAN, that is, the mitochondrial dysfunction interferes with the immune system in many different mitochondrial diseases that I treat. In fact, a relatively common cause of so-called common variable immune deficiency is simply a primary problem in mitochondrial function. And when you treat the mitochondria in the ways that people do these days, the common variable immune deficiency goes away. It has also been shown that there's a significant mitochondrial component in asthma, in that there's an association of certain mitochondrial haplotypes with the severity of asthma. So the question, so yes, treating there are probably will be many ways to treat MEPAN, but the but the question is, what is your mitochondrial treatment, or that is, you need to be seen by a specialist in mitochondrial treatment who can then look at all these problems. But and if it seems consistent with what we see in mitochondrial dysfunction, then simply making sure that you're on the best mitochondrial treatment will help, or should
Thank
help.
you so much.
Could I add a comment on that subject?
Mhm.
So that was the involvement of the immune system in primary mitochondrial disease has been like one of the largest areas of growth in the past 5 years. And there have been some really interesting studies where they kind of tamp down the immune system in primary mitochondrial disease and seen very good results in the brain. And we would really like to understand that better. And we do see some signs of overactive immune system in our mouse model. And so we will be trying immune therapies on the mice. That's one of the things that we really want to try and see how that helps neurological symptoms. But it's really very early stages. But we're hearing a lot of anecdotal reports that people who have mitochondrial diseases also have immune complications.
Wonderful.
Did you get any specific reports regarding any MEPAN patients, Debbie? Because I don't see any autoimmune involvement in any of our patients. No.
No, we haven't seen any autoimmunity, but we're wondering if the problem might be the crossover of the immune system into the brain. And so it's kind of a secondary effect of having a mitochondrial disease. So one of the things the immune system does is it'll see mitochondrial DNA when it's in the wrong place and turn things on. And so we think that some of that might be happening in mitochondrial disease when there's not a pathogen. When you don't have anything to react to, it's reacting to dysfunctional mitochondria. And so that's separate from the mitochondrial dysfunction in the first place, and then a secondary effect. So maybe if we could stop some of that, the patients could do better.
Wonderful.
Thanks.
Miss Stephanie Howard, would you like to ask your question?
Yes, thank you very much. We are what you would call a MEPAN family. Our son, Alexander Howard, and Alex, I think you're on this webinar. Is a MEPAN patient. He was diagnosed, I want to say about 4 years ago, and we have been to the clinic in Portland with Dr. Hayflick. Alex at that time donated skin, blood, had several samples taken. In subsequent years, he also it was requested that he have more blood taken, and that was sent off also to Portland. My question is, Alex and I and our family want very much to participate in these studies, but do you have those samples? We were told that they would be shared with all researchers. If you do not have those samples, what can we do to assist you in your research to get you those samples?
I know for my case, I primarily would like human blood to get PBMCs. And as far as I am aware, there's none currently available. I know my lab has gone through Stanford's program where a previous family has also donated blood, and we have an MTA agreement. So if anybody wants to go through them, that's a possible way where we can get samples from.
I'm curious if this might be a place where the UMDF could be more involved, because they are a central repository for patient samples. So is there somebody from UMDF that could comment on that?
This is Richard. I would say that this might be a perfect project for the Coriell Institute, in that any physician can send a sample, just send the tube of blood to the Coriell Institute to have both lymphoblasts and DNA prepared, and the cells are available to anyone who contacts the Coriell Center. And for example, I banked about 20 samples of Barth syndrome there many years ago, and they've been used again and again. So ask your physician, look into submitting a sample for the Coriell Center. It doesn't have to be part of a major project. Yes, individual doctor send it.
If I may, this is Danny Miller. I actually have an agreement with a group at NCATS where if patients and families send their samples into the group at NCATS, they are willing to differentiate those cells, those fibroblasts, into iPSCs and have them stored at Coriell. So all I need to do is get, anyone who wants to do that, contact me after the call, and we can get that done and start that process. It's going to take about 6 to 8 months to differentiate some of those cells. But again, as Dr. Kelly said, those cells will be available to any researcher who is interested in studying MT-FAST dysfunction or MEPAN or, something related. So I'm in the process of doing that with Stanford with my son's cells. And Stephanie, if you are willing, you and Alex are willing to do that as well, we can get that going.
I was just going to quickly say that I think it would be awesome if UMDF had guidance for patients, at least if they're not in charge of centralizing it, because I can say I've gotten fibroblast lines from 6 to 7 patients right now, and it's not any one place. It's, individual geneticists or the UDN or other sources. And so I think, kind of having one repository would really be helpful for people.
Well, I want to make sure everyone's aware of NAMDAC, the North American Mitochondrial Disease Consortium, which is the biobank repository. For the UMDF ourselves, we have MitoShare, which is a place where people can upload their genetic information, which is another important thing for all families to do. Stand up and be counted, because as we have these new developments coming, we want to know where people are and be able to get you where you're going. We could have people that have MEPAN in our database that we don't even know have MEPAN, because there hasn't been a place for that kind of information. So I do encourage everyone to join MitoShare, and your contact there is Miss Nicole Wilson, who is on with us today. Nicole, go ahead and put your information into the chat so that everyone knows how to reach you.
Absolutely.
I think that would be a great thing for all families impacted by MEPAN to do.
Great, thank you. Definitely MitoShare is going to be very important, but also Coriell is important. And I think both could be very helpful in coordinating some of these efforts, which is why we're having this meeting. Leslie Zent, did you have another question? I guess your hand is just still raised. Are there any other questions from the family that we can help with? Well, I think this has really been a fabulous effort, and I really want to thank UMDF for organizing it. I would like to invite Sherry Breslow and Dan Miller to make any final comments, as they have certainly been leaders in bringing this disease to the forefront and bring us all together to help solve this important problem for their children. Sherry, did you want to mention something?
Yes, I just want to once again thank everybody for joining us today. I mean, everybody collaborating together with all the different amazing research that we heard about today. We're going to get there. I know we are. So just thank you. And thank you again to Dr. Wallace and Dr. Murdoch, and for all the researchers. And thank you to UMDF for putting this together. And we're talking about getting the samples. So maybe after this call, we can go ahead and I know that all of the MEPAN families, I've sent out several emails with the link to the MitoShare. So I'll be glad to do that again to try to coordinate to get these blood samples together, because it seems like that would be something that the researchers would really want to have access to, and it's only going to help all of us.
Thank you very much. And Danny Miller, did you want to comment something as a co-organizer, co-host?
Yes, thank you. Thanks for so much for putting this together and for all the talks and questions. I just want to acknowledge some of the attendees on the call. Sa is from Dr. Hayflick's lab. Bashiar is from University UCal San Diego. They are looking at the acyl carrier protein involved in MTFAS. And then there were some other folks that had to drop off, unfortunately, but there are other groups doing more research on MTFAS and MEPAN, some of which the MEPAN Foundation and I'm supporting. So beyond what's been talked about on this call, there is additional work. There's another participant, Rima, from the University of Florida, who has started work on the gene therapy that was mentioned. Dr. Heimer talked about the gene therapy being possibly very amenable to that MEPAN may be very amenable to a gene therapy, and that work has begun, and we are supporting that, and hopefully we can use these mouse models and collaborate and work together to advance the notion of a gene therapy and test that. It'll take it's a long, complex process and will take a while, but we, as Sherry said, I think we can get there. We just need to keep talking, keep sharing data, and stay in contact. So thanks, everybody.
Thank you very much, both the Breslows and the Millers, for their dedication. Would any of the panelists like to make a final comment, or would Dr. Gailey Heimer and Richard Kelly make a final comment before we thank UMDF and sign off?
Go ahead.
Thank you, Doug. So my video is, I don't know, it's muted also. So if you can unlock me, but if not, you can see my husband. Just there was a question in the there was a question in the chat regarding the clinical variability between the patients, and actually we don't really know the reason for that. It's not only the different mutations, because as I said, all our 9 patients have the same mutations, and actually some are even siblings, so they share other parts of their DNA as well, and we see great variability in these patients. We can't explain it. I think it's, well, there might be modifying factors, but also it can be related to the fact that mitochondrial disorder is a stochastic disorder, and it can be affected by the occurrence of various metabolic crises, which might have influenced some and have not influenced others. So I don't have a good explanation for that. I don't know what you guys, Doug and Kelly and Dr. Kelly, think about it. Do you have any insight for the variability between patients?
Yeah, for the variability, it's genetics.
Yeah. Well,
the obviously it could be that there are many genetic factors. It's normal for there to be variability with the same mutation in the same family. That's not a surprising thing. If you had something that was completely in left field, if for example a person in the family, if a sibling was found to have MEPAN but absolutely no symptoms, then one would want to study that person in great depth. But the general variability in terms of what organs are involved, the rate of progression, that is influenced by so many different genetic factors, and it's just routine. And I treat mostly mitochondrial patients, or patients with some kind of mitochondrial dysfunction, and that's just typical. It's not that it should be ignored. As I said, there can be individual cases where one can learn a great deal. I'm less enthusiastic, although it could turn up something if you have enough patients to do genome screening to identify some common factor that the patients who don't have eye disease, although I guess they all do. See what I'm saying? That you could take a group of patients and look for something, but in most cases that is not very helpful because you get too many hits, and therefore, because I'm a clinician, I emphasize studying individual patients. But I hope that answers your question. I did have one suggestion myself. I was I looked at Dr. Heimer's mitochondrial treatment, and it's different than mine in some respects, and I can see things that could be improved, and maybe something that could be improved in my treatment as well. But because I treat so many different types of mitochondrial diseases, probably 50, and I've tried, and I have and I don't think any one patient is on the same treatment. I think it would be useful to have a separate meeting of the clinicians to talk about what they are using, why they're using it, and what the evidence for benefit is. I think it's not that everyone has to use the same treatment, but for example, something that's not generally known is that a major cause of progression in mitochondrial diseases is a deficiency of methionine or a deficiency of serine. And many people just aren't looking at the amino acids. So I think there's a lot of progress to be made in pooling our expertise or our experience with mitochondrial treatment to come up with some recommendations for MEPAN patients. Most neurologists are not trained in devising mitochondrial treatments, and so my experience has been that patients with the same diagnosis are on all sorts of treatments. And one principle I emphasize is that antioxidant treatment is not the whole answer. It's only a small part of the answer in treatment. So I think that's a worthy area of collaboration.
That would be great. Thank you.
Okay, that very much addresses Ms. Stephanie Howard's question about specific guidance for next steps. We definitely need to have this effort go forward. Perhaps UMDF and the Breslows and the Millers will help to think about another meeting where we can advance this information. There is one comment from Shu Yun Jun, and it says, us at NBIA Cure at Oregon's Health Sciences Center maintain NBIA repository with primary samples, and we are open to share any samples with any researcher in the world. So there's a third repository that we can now count on. Again, I want to thank UMDF, the Breslows, and the Millers for really championing this cause. And we're all dedicated to finding a cure. I've been in this business 50 years, and I still hope we'll find a cure for primary mitochondrial disease. I'm not giving up. I hope you don't either. So have a great day.
Thank you, Dr. Wallace, and thank you to all of our amazing panelists today.